ma'am, we are live on our YouTube channel. So, yes, ma'am, so that would be great if we can start the event. Hello, am I audible? Hello, hello.
Yes, you are.
Yeah, yes, ma'am, we are live now, and now we can start the event. So, good morning, everyone present over here on our YouTube live channel. So, we have come here to discuss upon the topic. Uh, the topic for the today is a biophysics topic, which says that the guiding minds and resolving mysteries, biophysical approach to molecular functions. Yeah, quite interesting with the name. So, yeah. So, ma'am, we have a really very nice biophysicist, eminent biophysicist, Dr. Debjani Bagchi here with us, who is working as an assistant professor in the Maharaja Sajirao University of Vadodara in Department of Physics. So, ma'am, I am really thankful to you for providing your auspicious time out of your very busy schedule to us. Really, thank you, ma'am. So, moving forward to the event, I would like to invite Bhakti to introduce ma'am with her kind words. The stage is yours. Bhakti, can you please introduce? Am I audible?
Yes, yes. So, good morning, everyone. Let me quickly introduce our today's speaker, Dr. Debjani Bagchi, an esteemed scholar and seasoned professional in her field. She has done her master's from IIT Kanpur, Uttar Pradesh, in the year 1999. She did her PhD from IIT Bangalore, Karnataka, while she was employed as a UGC project fellow during 2000 to 2007. She has done postdoctoral research from École Normale Supérieure Paris, sponsored by CNRS in 2012. She has published and edited several articles and papers in many renowned journals. Currently, Dr. Debjani Bagchi works as assistant professor at Department of Physics, Faculty of Science at the Maharaja Sajirao University of Baroda. Her research interest includes studying dynamics of soft and living matter, applications of soft matter in photonics, bioenergy, and renewable sources of energy. Her expertise lies in dynamic light scattering, X-ray diffraction, nanomaterials, soft materials, polymers, colloid dispersion, rheology, biophysics, and X-ray scattering. Her ongoing research topic is using lignocellulosic biomass for microbial growth to produce useful products like chlorophylls and carotenoids. Other research projects are out of equilibrium dynamics in polymers near glass transition and dynamics of out of equilibrium systems and experimental study. Ma'am, we are very pleasure to have you as a speaker today. And now, with this warm welcome, I request the host to continue the event.
Yeah.
Thank you.
Thank you, Bhakti, for your such a nice introduction for ma'am. I'm also really thankful to you again for coming as ma'am. So can we please start with the event with the question answer session?
Thank you very much, Soumilya and Bhakti. So
Yeah.
just let me know how should I start.
Yes, ma'am. The very first question with the which we have for you, ma'am. Can you please share a moment in your research journey that was particularly challenging or rewarding, and how it shaped your career?
Uh, so challenges are like just a part and parcel of this research journey. So the more the challenges, the more I feel, the more fruitful it becomes. Uh, Something very easy to solve, anyone can solve it, and the more the challenging the problem, the more interesting it becomes. For me, the first round of challenges started when I had joined my PhD at IISc Bangalore. And I guess in my second year of my PhD, I lost my advisor. Uh, in a road accident, and that was like a very sudden loss for all of us. And we had to come to terms with it. And it took us some time to come to terms with the loss. And then I had my CSIR fellowship, and with the help of the fellowships contingency and with the help of the department, uh, I could actually do my projects, think about something different. So first, thinking about what to do was the real challenge, because now there was nobody to guide. And then you find that when you find that you are completely on your own now, so then that is the time we become more responsible, because otherwise we put a lot of responsibility on the guide. Okay, sir or madam is there, so they will show
Yeah.
us out of the way. So if there is any problem, we can of course approach them. But now there was nobody, and of course there was nobody in that field working in the department. So like all of us in the lab were on our own, and very inexperienced. So that was a real challenge. And um, that was the time I used to attend a lot of courses. I used to read a lot to deepen my knowledge of the field which I was working on. I was working on complex fluids, and the physics of complex fluids is very challenging actually. And the last year, the Nobel Prize was awarded to the physics of complex systems, the Nobel Prize in Physics. And so that was like a very wow moment for all of us. That time when I was working on these complex systems, the first important thing was to first understand about the dynamics in these systems, and then develop a problem which can be like a research project. And so from then on the journey started, and I started liking this research field a lot. Of course, the challenges were there, and there were setbacks, plenty of setbacks, many of the things which I thought didn't work out. And yeah, so many things didn't work out, but some things did work out. And that was, whenever something would work out, that would be the wow moment.
Yeah.
And
Like
yeah,
a eureka
then
moment
I
it would be.
could, right. And so I could publish some papers, and I could finish my PhD. And then I remember there was a conference on statistical physics there, where I met my future postdoctoral advisor. And that was Professor Sergio Ciliberto. He was at École Normale Supérieure in France, and he was working on out of equilibrium dynamics. And so we discussed a little, and then when he had an open position, I got that position. So then I went to France. And but the thing is, when I was doing my PhD at IISc Bangalore, I used to listen to a lot of talks, interdepartmental talks, interdisciplinary talks within the department talks, and that also interested me to biological systems. So most of these biological systems, the dynamics in them are out of equilibrium dynamics. And when I went to France, that actually deepened my interest more, because there were a lot of people working on those systems, talking about them. And as you see, the challenges have actually shaped my research and my interest. And I feel that because of the challenges, I am still interested in research.
Yeah. Yes, ma'am. That we can
Yeah,
see
otherwise,
from your motivation and being a student of yours in during my internship, I saw the spark of research in yeah. Ma'am, you said that you have done your MSc and Masters from IIT Kanpur. So, as we all know that very renowned professor, Dr. Professor H.C. Verma, he's also from IIT Kanpur. So, ma'am, can you please share some memories with Professor H.C. Verma? What is his aptitude and how did you like him and your journey with him?
okay. So I was very fortunate to be at IIT Kanpur at that time. We had like some of the best teachers in physics. Uh, and of course, Professor H.C. Verma was one of them. And initially, when I had joined, then Professor H.C. Verma used to teach us mechanics, classical mechanics. And the mechanics wasn't so such an interesting subject for me, but when I started learning from HC Verma sir, it was different. So it made us, because that the way he taught, it made us think about different aspects of the dynamics of rigid bodies and different mechanics topics. And he would make us solve different types of problems. He would himself make the problems of the assignments and give us assignments to solve. And of course, the assignments were an integral part of the IIT Kanpur syllabus, and we used to solve and return back the solution, the solved assignments. And another thing was there that he would take quizzes in the class. So uh, and sometimes if the solution strikes you, you will get a 10 out of 10. Otherwise you would get a 0. And I remember for the first time in my life I had got a 0 in
Oh,
one of the quizzes. And yeah, so I was like, how can it happen? It was a little bit a good reasonably heavy setback, because I had never scored 0 anytime in my life. And so was that actually uh, it actually inspired me to actually imagine that what the imagination, the way in which I was thinking wasn't correct. So I have to improve my thinking, or I have to improve my understanding. And I feel that setbacks are important, and that taught me an important lesson. And um, I used to be a little nervous as to how to approach, but then I learned how to do it. And Professor H.C. Verma was very helping, very kind and helping. So whenever we used to approach him with some problems whose solutions we were not able to understand, he would give us hints as to how to solve those things and those problems. And that actually helped us a lot. We used to many times, I remember, we used to go to one hostel, all of us to a lot of us together, and we used to solve the assignments together. And so solving Professor H.C. Verma's assignments were really very interesting for us. That was like very nice brainstorming. And I would say that because of that, the knowledge of mechanics, which we all developed, was really very profound, because of his way of teaching, his way of imparting knowledge to the students. Um, I
yeah.
feel that is a very nice. That was a very nice way which he followed, and was a very wonderful teacher also. And he used to work on nuclear physics.
No.
So I remember that when it was time for our project, we had a few of us. We had gone and approached him that can we about his work, to learn more about his work on nuclear physics. uh, for like, because for our projects we had to go to different professors and approach them, learn about their work. And that was the first time I learned that about his work on nuclear physics as well. Um, and it was really very interesting, because being students, we were like not knowing his other dimension in research. And that was a very eye-opening, interesting moment for us, that sir not only teaches but he also does some very interesting research.
Yes, ma'am. Yeah, I feel that HC Verma is quite, he thinks really out of the box. And also, he advises students to think out of the box, which is really very great for it. So, ma'am, moving on the further, ma'am, innovations often requires to thinking out of the box only. So, ma'am, how do you encourage creativity and innovative thinking in your research?
Okay, so as I said, as you correctly said, creativity, out of the box thinking is the most important, uh, or rather, I would say it is the prerequisite for good scientific research. And I started right from my classes. So, I see that students many time they ask for notes. Ma'am, give your notes or give some notes to learn about this subject. The thing is, first I start. I don't give them notes. I teach them in the class. I tell them about different places where they can read from, reference books or some other places where they can read and improve their understanding. Then that actually, because I feel that when you give them notes, they just reproduce that and learn that, and their knowledge is restricted to that notes. And so that is what I dislike. And that doesn't actually improve the students abilities of on creative thinking and thinking out of the box. Because by learning on his or her own, a student can actually develop a better understanding of the particular subject which the student is working on or learning about. Not the notes are not good. And um, and I do also tell students to make some small projects at the end, like this semester I am teaching thermodynamics. I have told students to so thermodynamics in cell and molecular biology. uh, so I had told students to think about some small projects where they can apply thermodynamics to solve cell and molecular biology problems. So they will think about their own problems, they will solve it, and then let's see what type of
Yes.
problems they think about and how they solve.
Yeah,
So
ma'am.
that would be, I felt interesting. Once I had made them, I had given them some projects, some problems, and told them to do that. Uh,
Yeah.
so I follow different approaches from time to time. I remember I had made them give presentations on the subjects of the subjects, the topics which I was teaching. So presentations on the applications of the topics. Sometimes I do have group discussions also. So that way I try to develop some creativity, because suppose if the student is interested in that particular field, the student is also creative in that domain. So interest is another important thing. And the most challenging thing is to keep the students interested in what what I would have to teach, what I am assigned to teach in the class. Yeah.
Yeah, yeah. I've got the opportunity to learn from you in my bachelor's, and I was really very fascinated with the kind of teaching which you provided to me and guided to me. Yes, ma'am, moving on further on your research, which is on soft matter, and so ma'am, soft matter seems to interact with many disciplines. So from photonics to renewable energy. So ma'am, how do you see the potential of this field evolving in the next decade? And further, can you please shed some light on soft matter physics to our audience who are from various backgrounds from botany, biology, who can really understand it from your kind words?
Okay. So, yeah, as you correctly said, soft matter physics encompasses a lot of domains in the different sciences, not only in physics. First of all, biological matter is soft matter. So physicists look at biological systems as soft matter. A cell is like a soft matter with its own mechanical properties. The best part of these soft matter systems is the experiments are so simple that they are doable with simple experimental setups in the lab. And the most important thing about these materials is if you uh, if you subject them to a small perturbation, the response of the system is really very interesting. So suppose if you have a cell and you subject it to some mechanical uh, uh, variations. Suppose the cells are adhered to a substrate, and you subject and you make this substrate of different mechanical rigidity and study the cell migration. Then you will see that the response which you are studying, which is the cell migration, is different for different substrates, and that helps us in looking and studying the metastasis which happens in cancerous tumor cells. Okay, and so you are linking some mechanical properties to the metastasis. So basically, you are linking some physical phenomena to some important biological phenomena which is happening. The same thing when you are studying drug delivery, for example. Again, the mechanical property here is the diffusion, where the drug has to diffuse to the substrate, or to the wound, or to the place where it has to go. So again, the micro scale mechanics comes into the picture, and the mechanical properties of this living matter is completely different compared to the mechanical properties of the matter which you have in day to day life, and each has their own unique feature. So for example, if you take a tube of toothpaste, it is designed in such a way so that if you squeeze the toothpaste, it will flow. Otherwise, it is like a solid thing inside the tube, and when you squeeze it, you tend to make it more flowy, or more resistance to the forces which is causing it to less resistance to the forces which is causing it to flow. So there is a transition from a gel to a fluid like state in the toothpaste. So the toothpaste has that particular property. In cells, there is non-affine behavior when you subject it to mechanical perturbation. So the actin and microtubules, when you subject them to mechanical perturbation, uh, their mechanical properties are very heterogeneous and very interesting to study, and that has been observed to play very important roles in various cellular processes. So I believe that first of all, these simple yet very powerful things can't be neglected. So in future, a lot of research would be directed towards this interdisciplinary understanding, wherein one has to combine biology with chemistry and physics, and as well as mathematics as well.
Yes, ma'am.
So uh, right now, there is a very interesting branch of research where the mathematical principles like topology are applied to DNA packing or knots in biological systems. So, mm, so it's not only physics but mathematics, statistics, chemistry, everything would be together in the future to actually help us solve problems in a better way.
Yeah, really, it's a, it is a thought provoking topic for me. I have never think into this direction. Yeah. So can we, is it possible that we can detect some with the help of soft matter physics? Can we cure the disease which don't have the cure like prion disease or the neurodegenerative disease? Can we get some information for that using soft matter physics? Right.
Yeah. So there is an important branch of soft matter physics where we look at phase transitions. So phase transitions, as you see, even in day-to-day life, the transition from the liquid to the gaseous phase, or from the solid to the liquid phase, that is very common. But there is a transition called liquid-liquid phase transition, wherein from a mixed state, the liquid demixes and forms domains of its own, and these transitions are seen in cells. And now for the first so this is an important part of soft matter physics. Most of my PhD thesis was centered on this liquid phase transitions, and for the first time now people have found out that the very it had been very challenging to actually find out the very initial stage of Parkinson's or Alzheimer's disease, where there are these aggregations of some proteins forming fibrils and finally causing neurodegeneration. And people were it was very challenging to find the initial stage of the disease, because the disease would be detected when the disease had progressed to a very large extent and had damaged a lot of neurons. And then from then on it wouldn't be curable at all. So very recently some soft matter biological physicists have actually found out that in the very initial stages of this disease, the prions or the proteins start forming condensates in the cell by liquid phase separation. And there are small protein condensates in the cell which can be detected in the very initial stages of the disease. And these condensates initiate the process of oligomerization of the proteins, and then first aggregation, because the proteins start aggregating within these condensates. When they aggregate, they oligomerize, and then they form fibrils and so on. So that was a remarkable achievement of soft matter physics. And I believe that this would soon be translated to some better diagnostics in the future. So, translation research is a little bit still behind, lagging behind compared to the actual lab bench research. So I believe a lot of translation also has to be employed, and clinical studies should go together with these kind of discoveries which is happening. So that was an important discovery recently. There are many more, of course, and
And it was a
so
really completely a new information which I am getting. I hope it will be a new information to our audience also, because I never came across such things that this phase transition also happens in neurodegenerative disorders as well. So, ma'am, uh, when I was looking for your research papers, I found that in your research papers you are quietly working actively on chlamydomonas renardi and Raman's spectroscopy. So, ma'am, can you please shed some light on it? What is actually how a kind of physics a technique is used in a biological model and how spectroscopy is used in this such kind of things, and so that our
Okay.
audience can get to learn more.
Okay, so recently that has been my favorite subject. I have been working a lot on this domain, and so what I found out was when I was working with the biochemists and molecular biologists, of course, and phycologists who are studying microalgal systems, I found something. I had actually worked also in one of the labs, leading microalgae lab. So uh, I found that to the microalgae, first of all, the systems were very interesting. Various microalgae, like Dunalella, was very good at carotenoids production. Then microalgae are harvested for omega 3 fatty acids as well as lipids, which have been used for biodiesel. And microalgae, that's why interested me a lot, like they are like mini factories of nature. And I started studying them a little bit more, and when I was working in the lab, I found out that biochemists have very nice, well developed protocols for extracting these molecules from microalgae. And of course those protocols were there, but suppose you want to study the microalgae, the molecules within the microalgae before extracting them, then they didn't then that wasn't possible. And another thing is, in order to study the features of the molecules, you had to use a lot of microalgae, harvest them, and then extract it from a huge amount of so you need a good amount of starting point in the beginning. And you need lot of chemicals, lot of steps, extraction steps for these purposes, and then you would be able to finally study the molecule of your interest. And then I thought about that, why not study these molecules with some physics or some physical techniques. And that was when spectroscopy came into my mind, because that is a non-invasive technique. You shine light, you don't have to do any biochemical extractions. Suppose you want to optimize your conditions, you don't have to take a lot of system to start with. You don't have to spend a lot of chemicals or do lot of steps. You just have to shine light on a small amount, uh, or maybe a single cell or even some few microliters of your um, culture. So, when you shine light, what happens? Each molecule has its own spectral fingerprint. So, the spectrum is like a fingerprint of a molecule. So, like each of us have our own fingerprints, which is distinct to us. The spectra of each molecule is distinct to that particular molecule. So, lipids have their own fingerprints, carotenoids have their own fingerprints. And so that's what made me study these lipids and carotenoids a lot with Raman spectroscopy, and presently I'm studying them with infrared spectroscopy also. And of course, there are various other spectroscopic techniques which can be used complementary to each other, um, and extract as much information as possible. So, right now, I'm trying to understand, extract some information from these different techniques and try to model them with the help of artificial intelligence, to see that if there is something unknown in it, can the model actually give a layman what is there in the system. So a physicist will not always examine the systems with these techniques. So suppose a non-physicist examines, how will he or she understand about these systems? So the model should be sufficiently simple to give a output based on what is spectroscopically observed. So that is my current interest, and we are actively uh, looking at it.
Yes, ma'am. So, ma'am, adding further to this question, you said that how we can look the molecular aspect. So, ma'am, as you know, the biological system they keep on evolving and adapting to the current situation. So, ma'am, how spectroscopy can help us to detect the changes, the adaptation? How the can it be linked with the evolutionary kind of study? Is it possible?
Yes, yes, definitely. So spectroscopy can find out even the slightest signature of change which has occurred. For example, in one of our systems, we had subjected the cells to salinity stress, and what we found was that the carotenoids, which are secreted in response to this oxidative damage which happens in the cells, basically these are raw scavengers. And the carotenoids, what we found was they evolved from their configuration from one type of carotenoid to another type as the cells aged. So that was a simple thing which we had seen in the aging of the cells. One can apply this to evolutionary aspects also. So another thing which we saw was when you apply some stress to the cells are not behaving collectively. They are not showing the same behavior, in the sense that there are some subpopulations inside the culture which are showing behaviors which are different from the others, completely different behavior. So detection of the subpopulations becomes important, and that's how spectroscopy helps, because that time you are looking at the single cell level, and so you are able to detect these subpopulations. If you are looking at the whole thing collectively, then you would miss out on these subpopulations. So you would miss out on how the entire population actually develops mechanisms to cope with stress. Then what we found was there was something like division of labor which was occurring. Basically this development of different subpopulations is like something like division of labor, and actually that also helps the in analyzing the antibiotic resistance in bacteria. When you give antibiotics, then there are some bacteria which persist, which resist these antibiotics. They still remain alive. And how does that happen? When you look at the single cell level, then you see that there are subpopulations which are having different cellular mechanisms compared to some the entire population, and those are the ones which persist when there are when there is even in the presence of an antibiotic. So bacteria as well as these different microorganisms have can actually evolve and have very nice evolution mechanisms in response to stress, and that can be analyzed in great detail um, when you actually study them with these different tools like micro Raman spectroscopy and different other spectroscopic techniques, which you may not. So, if you want to study them with microscopy, you have to tag the molecule of interest. But here in these spectroscopic techniques, you don't have to do any pre preparation of your cells, and you don't need to tag them with anything fluorescent, any fluorescent tag. So you just have to shine some light on the cells, and then you get your answer out.
Yes, ma'am. Yeah, it's a really new dimension. So I was reading about some spectroscopic methods, and I got to know about single molecule spectroscopy. So, do you have any information regarding how single molecule spectroscopy could be applied to biological applications to combat the upcoming problems like antibiotic resistance and diabetes, the obesity, the lifestyle disorders? How can it be the single molecule spectroscopy can be used in that?
Okay, so I had used single molecule spectroscopy in my research when I was in France. So we were looking at oxidative DNA damage. So of course that is an also an outcome
Yeah,
of a bad lifestyle, but there are various other reasons of oxidative DNA damage. And when there is oxidative DNA damage, then there are various repair mechanisms which work. And I was looking at one such mechanism of DNA repair. And in single molecule spectroscopy, the best part is uh, you are studying the mechanism of a single molecule. So, of course, there are many proteins which are involved in the repair mechanism, but you take one protein at a time and you see how that actually interacts with the DNA or any nucleic acids. So I had looked at such protein nucleic acid interactions of many important proteins uh, involved in human diseases, uh, and we had looked at them in the single molecule level, and the activity of these proteins would tell us the difference between a healthy protein or a mutated protein. Okay, so the people whose proteins have certain mutations, then they are the ones which are most susceptible to cancer, okay, or aging induced damages. Otherwise, in healthy individuals, these damages are averted because of this repair DNA repair mechanisms. And once you know of these mechanisms at the single molecule level, you know exactly how a protein is working, how a protein attaches itself to DNA, how the protein repairs the damage, how the protein translates further, and yeah, so various aspects of his mechanisms are clear. So, what is the kinetics of ATP hydrolysis during this process, the dynamics of this molecules, the kinetic steps? Basically, you are able to see it with much more better resolution. Okay, so that is very important, I believe, for understanding for going into the basics of any disease in the first place, because that is the time when you have the different kinetic steps of the mechanism in front of you, and that is the time when you have those kinetic steps, you would be able to develop methods to actually not only diagnose the disease but also to develop therapeutics for the that particular disease.
yes, ma'am. Yeah, really, it's a mind-blowing topic to discuss. I feel that at least 5 to 10 PhDs can happen on such topics as well.
Yeah.
Yeah, yeah. So you talked about the kinetics. With the kinetics, I got a striking thing in my mind that are the molecular motors in our cell. So they are responsible for keeping our structure stability. What do you think about that? How molecular, if we study molecular motors properly in detail, so what can be possible outcome on that, and what novel thing we can find out from that with the help of soft matter physics and combining it with molecular motors.
Yeah. So molecular motors are very interesting because the thermodynamic efficiency is close to 100%. So if they hydrolyze, say, a few molecules of ATP, the energy obtained is almost 100% used for doing work compared to this the man-made motors. which work in about 20 to 30% efficiency. So first of all, soft matter can help us, actually has helped us to develop those nanobots or nanomotors, and people are actually working on, there are nano actuators developed, various nanobots which have been developed now. And I believe some chemists had got Nobel Prize in chemistry for developing um, molecular motors, molecules which were actually the nanomotors. Okay, so the chemists have actually designed various molecules which are actually also nanomotors. And these nanomotors have a very important role to play, first of all, in disease detection and disease diagnosis, because they can be very efficiently then they can be some receptors can be attached to them, so that they can actually be targeted to the site which needs to be examined, or where the drugs need to be injected to that particular site. So this is actually an important part of nanomedicine. And uh, uh, Here in this nanomedicine, these not only these nanomotors but quantum dots have also played an important role uh, to actually uh, track these nanomotors and study them in detail, if it is they are injected to cells or to some living organisms. um, I think I know one company uh, which actually develops these kind of nano needles to target cancer cells, and then inject drugs into the cancer cells and leave the healthy cells intact. This is the future of targeted and precise drug delivery and targeted nanomedicine, basically. And that way, the soft matter community can be of help in even in therapeutics.
Yes, ma'am. So, ma'am, I guess that all the three sciences, all the four sciences, the physics, chemistry, mathematics, and biology, when integrated, they can give a really blast ideas and so blast solution for all the problems. So, ma'am, but this kind of interdisciplinary approach require interdisciplinary research require a different kind of mentality and a different kind of approach. So, ma'am, since you also work on interdisciplinary work as well, so ma'am, what advice would you give to students interested in crossing these traditional boundaries in science and going to applying mathematics and biology or applying mathematics and physics and computers and biology? Ma'am, what advice would you give?
Okay, so actually, when I first when I first started working into this soft matter physics, I found that this domain actually involves physics, chemistry, biology, as well as, like, it's very interdisciplinary. And interdisciplinary domains are challenging, but they are very interesting, uh, in the sense that, suppose I don't have a very good knowledge of mathematics. Okay, but suppose I would need to apply a mathematical tool in my research, then I can actually read about that tool and understand, and then apply a particular thing to my research. So that open mindedness is important. And of course, if you are interested in that particular field, you would be able to read something different and apply it to your field. And that is very important, even if you are working in a restricted field. like say which is very much dealing with a particular subject. Even in a particular subject, you would have to read about different things and incorporate something new in your work. So this way, the interdisciplinary domains becomes very interesting, as well as very and because it is a little challenging, it becomes more interesting, and it gives way for more creativity. Um, and so you can it basically gives you a field where you can apply different knowledges together. It gives you a motivation to read and improve your knowledge on different things, in nature as well as in your lab. So it basically gives you an idea of correlating the natural observations with discoveries in your bench. And I believe that uh, in future you can't actually separate the disciplines, because I see now this Elon Musk had developed this Neuralink. His Neuralink company had developed this device, which was implanted in a brain of a human being who was actually not able to use the hands, had lost his ability to use hand. So that actually incorporated a lot of bioelectronics, a lot of physics, as well as biology. So as well as chemistry, because you need to design those components which are biocompatible. You need to develop the chemistry, and actually then you know the value of teamwork, because then you are working with a team of biologists, chemists, as well as physicists together. And then the problems become very interesting. You are not restricted to a particular domain.
Yeah, yes, ma'am. Really, yeah, we should explore these ideas as well. But as you all know that ma'am, there are some people who tend to they feel shy to interact with others and all. So ma'am, in the field of research, the networking and the collaborative approach I feel is one kind of a plus point if we have. So ma'am, according to you, how collaborations help in our research, especially in the kind of interdisciplinary area?
Yeah, collaborations can actually speed up research. It's not that you yourself can't do it on your own. You can definitely do it on your own. Yeah, you can even learn a little bit of mathematics. You can learn the mathematics which is necessary for you to apply it in your research. You can learn the statistics which is necessary. Okay, I could learn the computation which was necessary to do my artificial intelligence program. But suppose you collaborate with a mathematician, then the mathematician's input will be richer and it will be faster. And it's that it would be like a win-win situation where you as well as the mathematician would gain some important insights on a problem which was not previously which they hadn't previously thought would work. Okay, and when you see something which you hadn't thought would work, something which you had thought was impossible, then that makes your problem very interesting, basically. So working with collaborators, first of all, makes your problem interesting. It makes you solve the problems fast, and then of course it gives you brings you in touch with another team, and that way you can improve your networking. And of course the fourth thing is the excitement of solving a problem together, wherein it is not one but many people together which are trying to solve and understand a problem of interest. And yeah, I remember uh, so I was looking at this micro scale diffusion where we had developed these programs for analyzing them in different way. Various biologists had approached us to understand this program, and how to write this program, how to um, program from their perspective, because every system is different, every cell, every phenomena which they are observing is different. And many had approached me, and I had helped them out, and that actually makes it more interesting, because then you know that how your work can be applied by others. Okay.
Yes, ma'am.
And yeah, so even if it is not collaborative, but you can actually help them in a way in which they can apply this knowledge developed by you, so that will help in progressing the knowledge further. So whatever work you have done, that would be an application of the work done. So that is another way of working with people, if not collaboration. Okay. In both ways are very good. Okay. So I have worked in both ways, and I feel that nowadays doing single-handed work it makes it very slow. It's not impossible, but it's very slow. And if you collaborate with and develop a team of your own, then work is very fast and very interesting also.
Yes, ma'am. Yeah, yeah, you're really correct that collaborations really help us and it speeds up the research. It would be really great if we would have got a whole day to interact with you in physical mode. But ma'am, we have time limitations as well. So ma'am, we will be moving on to our YouTube questions, and they're really very interesting. So the very first question is from Kishore Ghosh. So ma'am, he asked, ma'am, what role does machine learning or AI play in biophysical research predicting molecular behaviors and structures?
Yeah, so both molecular properties as well as structures are an important part of understanding of various fundamental phenomena. And so artificial intelligence still has some limitations, but it can be intelligently, you can actually develop a model to encompass behavior of as well as the properties as well as the structure of a large subclass of molecules, and then analyze that particular subclass without having to actually do the experiments from because the model has been developed with experimental data which is present. And of course that will work only if you develop models with experimental data. And theoretical models are there, and they are also important to a certain extent, but experimental data can give rise to more powerful models.
Thank you, ma'am. Ma'am, the next question is from Anurag. He asked that, ma'am, what biophysical molecular techniques can be used in DNA repair mechanisms to study the DNA repair mechanism?
Okay, a lot of the techniques I have used single molecule force spectroscopy, where you use optical tweezers, magnetic tweezers, or atomic force microscopy to look at the single molecule events. You can use super resolution microscopy. You can use various quantitative fluorescence imaging techniques like FRET, okay, or FCS to study them. So these are some various single molecule techniques.
Okay. And the very next question is from again from Kishore Ghosh. He asked, how can interdisciplinary approaches combining biophysics with fields like genomics or systems biology can advance our understanding of the molecular functions?
Yeah. So systems biology, actually, if you see the pathways in a cell, there are several networks, and systems biology analyzes these networks, these different pathways, signaling pathways or metabolic pathways. So these are analyzed in a much uh, how to say, in a better framework where all are actually considered. So systems biology that way is very important. Um, and of course, otherwise analyzing these networks become very difficult. And many times you can't study so many metabolic fluxes, but of course there are several signaling mechanisms as well as metabolic fluxes which are also linked to each other. And without signal systems biology, the knowledge is incomplete in this way. And the other question was on what? Somalia?
on how it can advance our understanding of the molecular functions.
Okay, of the molecular functions. So, molecular functions, uh, one has to look at kinetics as well as the dynamics at the single molecule level. Then, understanding the biological molecules becomes easier because biological molecules are very complicated, very complex molecular systems with lot of interacting domains and various structural changes and multi-step kinetic processes. So, looking at the single molecule level becomes very important then, and that's why there is a spurt in this single molecule biophysics research.
Yes, ma'am. Thank you, ma'am. The very next question is that what are the limitations of biophysical methods when studying molecular functions, and how can these be over?
Yeah, so the major limitation of these biophysical techniques is they are much more easier if you do them in vitro. And in vivo, it's very difficult, but it is not impossible. And so the research is still in progress, but people are developing methods uh, to look at these biophysical processes in vivo. Mostly they are fluorescent techniques, but now single molecule force in vivo assays have also been designed.
Okay, ma'am. Yes, ma'am. So, uh, apart from the academic point of view, there are many students who are doing PhD, and they are into more interested into research, but they try to feel that they feel depressed when their results don't come, and things don't work how they want the things to work, and there are some kinds that some challenges, administrative challenges are there. In that also they feel depressed that why I am doing PhD. So ma'am, how you can motivate these young researchers who are going to be budding scientists in the upcoming future?
Okay. So failures are very important, and they help us learn. That has been my very important lesson, which I have learned in my research career. Without my failures, I wouldn't have been what I am today, or I wouldn't have been able to develop any research field further. And the more challenging the field is, the more are the failures you encounter. And if you fail, that is correct. Yeah, you have failed. I mean, you have to take a lesson out of it and do something which which actually improves the mistake which has been done. Or if it is not a mistake, you know that particular system is not working. Now you have to think about something else, and that is very important. And because that sharpens our understanding, that sharpens our thinking, and that actually failures should motivate us rather than making us depressed. And repeated failures, I have seen people having repeated failures. Just a second, I have to open my door. Yeah. So actually, I'm sorry for this interruption. So there are people which have faced repeated failures, and there has been a friend of mine. She was actually trying to crystallize these membrane proteins, very important class of membrane proteins, and that was a real challenge for her. And she had encountered huge number of failures every year. Okay. And I think at the end of her fifth year, she got the structure which was so important that it could be published in Nature. And so that persistence in the face of failures is also important if you are if you want to do something new. And if you see our Nobel laureate Professor uh,
Thank you,
Venki
Ram.
Ramakrishnan, so don't you think he had faced a huge number of failures before he could actually
I
study the
feel
structure of
yes.
those ribosomes, isn't it? So the
Yes.
more challenging thing you want to do, the more the failures you will face. But each failure should make you think about a strategy to improve, or it would tell you which way you should study, or you should whether you should abandon the subject and move on to something else. But quitting should not be an option, because you should know in whatever you want to do there would be failures. Okay.
Yes, ma'am.
And of course, simple things you can be successful very fast. So you can keep, to be on the safe side, some simple things also, and some challenging things also. I believe that both problems, if you do to to actually keep you motivated, something very simple which makes you very successful fast, and something which is very challenging. So if you do both, then it would be good actually to keep you motivated if you lose your motivation. So that is another way.
Yes, ma'am. I'm really thank you for providing us with such a insightful talk about this event and about the new things which you have given us. But really, we don't have now. We have to be end our event because we don't have all the day. But it would be really great if we can get some kind of offline interaction and workshop with you. With this ending session, I would like to invite Vaishnavi to conclude our session and provide a vote of thanks for our audience. Vaishnavi, are you there?
There and yeah, yeah. Am I audible?
Yes, yes.
Yeah, yeah. Okay. Yeah. Thank you very much, ma'am, for coming to this event and joining us for this very beautiful morning. Your journey and your has inspired all of us and motivated us. Thank you very much for doing this, and thank you to all the audience who joined today. And yeah, ma'am, we would like to have another, as Soumya said, some offline interaction with you as well because it was very interesting. I learned many things apart from what you teach in the class and what is there in the course. So, yeah, it has developed my interest as well in physics a bit more. And yeah, thank you very much, ma'am.
Thank you so much for organizing this program, and it was very interesting. And I hope that more students take advantage and
Yes, ma'am.
get motivated with
Yes,
this.
ma'am.
It's a very nice initiative.
Thank you
Thank
so
you so much,
Thank
all
you,
the
ma'am.
organizers.
Yes, ma'am. To the all audience, I would
Thank you very
say
much,
yes,
ma'am.
ma'am. To
Yeah.
the all
Thank
audience, I would say that we have posted our feedback form in your comment box, so you can please look into it and fill it if possible, and so that you can get the e-certificate from it. So please don't forget to fill up the feedback form, which has been shared in the comment box. So please do fill up the feedback form so that you can get the e-certificate, and to please do like, share, and subscribe our YouTube channel so that we can come up with such more insightful talks. And I'm really thankful to you, ma'am, for providing your time on such a auspicious day and a busy day as well. Yes, ma'am. Really thank you to you. Thank you so much, ma'am.
thank you. Bye.
But yes, ma'am. Bye, ma'am.
Yeah.
So bye everyone, and happy Ganesh Chaturthi to all the audience. Bye bye. And please do fill up the feedback form in the from the given link in the comment box. And thank you. Please do like, share, and subscribe us again. And don't forget to hit the bell icon. Till then, think, share, and grow.